really like to express my gratitude to the Sjöberg Foundation, the prize committee, as well as the Royal Swedish Academy of Sciences and the Karolinska Institute for selecting me for this award. It's certainly been an impactful award for me personally, but also the team of scientists who have worked with me in my laboratory, as well as various collaborators in terms of some of the discoveries that I'll share with you today. Certainly this year is particularly special, coming post-pandemic, as well as having a triple header of speakers at this particular Sjöberg Foundation event. And I'm certainly quite grateful that the foundation was very welcoming of us and our family here in Stockholm as well. So what I thought I would do today is begin a bit historical in terms of the discovery of these recurrent gene fusions in prostate cancer and in common solid tumors, how that has been translated into other epithelial tumors, how various diagnostics have been developed from this work, and move on to a more recent story where we've attempted to indirectly therapeutically target the products of this particular gene fusion, which are actually oncogenic transcription factors, which, as you heard from other speakers, have been notoriously difficult to target with small molecule inhibitors. So just to give you a bit of background, especially for the students perhaps in the audience, and to sort of set the stage in terms of these gene fusions, the molecular basis of chromosomal alterations in terms of cancer really goes back a long way, back to Theodore Boveri, who first hypothesized a causal role for chromosomal aberrations in cancer. And when we're talking about these alterations, we're talking about duplications, amplifications, insertions, deletions, translocations, and gene fusions. However, before our particular study came out, it was really thought that gene fusions and translocations were the molecular basis of the liquid tumors or the hematologic malignancies, and generally not found in common solid tumors or epithelial carcinomas. But once they're actually identified, I think what's intriguing is that they actually end up being the driver of that particular cancer type, especially if they're recurrent in nature and essentially causal for that cancer type and can serve as a therapeutic target. And perhaps the poster child for this you heard about already was really the Philadelphia chromosome, work by Hungerford and Noel in terms of identifying this, and the BCR-ABL gene fusion, which characterizes this form of leukemia as a translocation that creates this gene fusion. But the beauty of this is that this gene fusion can be targeted with small molecule inhibitors, such as the ABL kinase inhibitor Gleevec or imatinib. And patients that have been given this drug have certainly had quite significant responses, and thus this has really been one of the, again, hallmarks in terms of precision therapy for cancer in general. A little bit of background in terms of prostate cancer in general. Our work really builds upon some of the pioneering work in terms of androgen signaling in terms of prostate. And in normal development of the prostate, as well as in prostate cancer development, it's known that the male hormone or androgen signaling is important in the growth of the normal prostate as well as in prostate cancer. And Charles Huggins received the Nobel Prize in 1966 for discovering that one could block hormonal signaling or androgen signaling, and this could be a treatment for prostate cancer. So androgen or testosterone binds to the androgen receptor, which is a transcription factor. This transcription factor dimerizes, gets translocated to the nucleus and drives target expression. And so this is where the gene fusion that we identified sort of comes into play, in that what happens with these gene fusions is that these oncogenic transcription factors, ERG being the most common, hijacks the gene the TMPRSS2 genes enhancer regulatory network such that this is a testosterone or androgen-regulated gene leading to overexpression of this oncogenic transcription factor. And as you had heard earlier, this was certainly a shocking observation at the time because it was really thought that these gene fusions or translocations were the cause of hematologic malignancies, and we had identified this in a common solid tumor where basically you have male hormone through the enhancer regulatory network of the TMPRSS2 gene drives ectopic overexpression of these oncogenic transcription factors in prostate epithelia to drive prostate cancer development. And this is the structure of the first two gene fusions that we identified in prostate cancer, TMPRSS2 ERG and TMPRSS2 ETV1. So how did we make this discovery? So this was actually during the time pre-availability of next-generation sequencing-based technologies. This is really the time of cDNA microarrays. This is before the TCGA efforts as well as the ICGC efforts in terms of sequencing tumors and so forth. And we weren't actually looking for gene fusions or translocations in prostate cancer per se, but we had developed a bioinformatics approach, or we developed an outlier approach to nominate genes that had very high expression in a subset of cancers, most cancers not necessarily having expression, and low expression in benign tissue. We applied that outlier algorithm to a cancer microarray database that we had assembled called Oncomine, where we assembled at the time the world's microarray profiling data across different cancer types. This outlier algorithm nominated ERG as well as ETV1 in prostate cancer, and we went on to carry out exon walk PCR and noticed a disbalance in terms of exonal expression, whereby the 3' exons of ERG, for example, had very high expression, while the 5' exons had attenuated expression. And we went on to use conventional technologies to sequence the 5' end of these transcripts. So we used 5' race, and we were shocked at the time to really find fusions of TMPRSS2 to ERG and TMPRSS2 to ETV1. We ended up using multiple different orthogonal approaches to try to validate these findings, including fluorescence in situ hybridization, as well as other approaches. Because prostate cancer is so common, and this gene fusion, and in particular the TMPRSS2 ERG gene fusion, is represented in about 50% of prostate cancer. This ends up actually making TMPRSS2 ERG the most common gene fusion in cancer, or common recurrent gene fusion in cancer. It's being utilized in pathology labs as a very specific biomarker of the disease. Here I'm showing a patient that's been stained for the ERG gene fusion product, being positive. This particular patient is positive for the TMPRSS2 ERG gene fusion. So since our discovery, certainly these recurrent gene fusions have been identified across other epithelial tumors, including in breast cancer, various colon cancer, ovarian cancer, as well as in lung cancer. And perhaps the most intriguing observation was the discovery of the EML4 ALK gene fusion in lung cancer by Hiro Mano in Japan. And the reason that was so exciting at the time was because that particular fusion can also be therapeutically targeted with small molecule inhibitors. We went on, as the emergence of next-generation sequencing-based technologies and transcriptome sequencing technologies became available, we began to develop the first approaches to actually use transcriptome sequencing to discover gene fusions, and we developed variations of this that could be used in degraded samples, formalin-fixed paraffin-embedded samples, such that one could identify and discover gene fusions across different sample types. And in prostate cancer, we ended up actually finding many different flavors of gene fusions actually in prostate cancer, although TMPRSS2 ERG by far ends up being the most common. Different ETS transcription factors create gene fusions, but we also found some other interesting gene fusions that represent rare subtypes of prostate cancer, such as those that are made up of RAF fusions, BRAF as well as RAF1, R-spondin fusions, PI3 kinase fusions, as well as FGFR2 kinase fusions. And what's interesting about these recurrent gene fusions in prostate cancers is that they're mutually exclusive, and that if you have an ETS fusion, you tend not to have, for example, a RAF fusion or an FGFR kinase fusions. These gene fusions are generally early events in prostate cancer development. They're truncal events that occur during when precursor lesions transition into invasive disease, and generally they're clonal events in that all of the cells will have these recurrent fusions. Our group and others have shown that when we express these gene fusion products in mice, they develop precursor lesions of prostate related to prostate cancer. Charles Sawyers went on to show basically that when mice were crossed with loss of PTEN and TMPRSS2 ERG overexpression in the mice, that these mice develop prostate cancer at a relatively rapid pace. And just mere introduction of the gene fusion in mice and aging these mice over a period of time, the group at the Fred Hutchinson Cancer Center has shown that these mice develop prostate cancer at a high penetrance. And this actually aligns quite nicely with what we see in humans, where prostate cancer is generally found in older men. So these mice, when aged to about 2 years of age, they develop prostate cancer with just mere introduction of ERG overexpression under androgen regulation in the prostate. As I had mentioned, these gene fusions are exquisitely specific for prostate cancer, and thus we've been able to take advantage of the TMPRSS2 ERG gene fusion as well as the long non-coding RNA PCA3 as very specific biomarkers of prostate cancer that we can detect in the urine of men with prostate cancer. And what's interesting is that these products detect high-grade versions of the disease and can then prevent unnecessary biopsy with these tests. And this is just a graph showing that generally high-grade prostate cancer patients have higher levels of TMPRSS2 ERG and PCA3 relative to low-grade versions of prostate cancer. This is just showing that PSA, or serum PSA, which is commonly used as a screening test for prostate cancer, ends up not being able to distinguish low-grade and high-grade prostate cancer, while TMPRSS2 ERG and PCA3 can distinguish these types of prostate cancer. And this is actually ends up being one of the most highly validated urine biomarkers for prostate cancer, TMPRSS2 ERG and PCA3, as it's been validated by many independent groups. So in terms of these discovery approaches for gene fusions, as I had mentioned, we had discovered or we had utilized transcriptome-based sequencing-based approaches to discover gene fusions. We felt that we could potentially transition these sequencing-based approaches into the context of precision oncology. So we developed the Michigan Oncology sequencing program, or the MyOncoSeq program, where we essentially combined RNA sequencing with whole exome sequencing of tumor matched with normal, as well as whole genome sequencing that we could deploy or utilize in patients with advanced or metastatic cancers. And so this is actually one of the first of its kind comprehensive cancer sequencing, clinical cancer sequencing programs, whereby we, again, as I mentioned earlier, carry out a tumor biopsy, generally of metastatic disease, compare that to a matched normal, a germline specimen, carry out integrative sequencing, analysis, presentation at precision medicine tumor boards, and disclosure to patients. So in 2010, this was one of the first of its kind clinical sequencing program that was really based on some of the technologies that we were using to discover gene fusions and characterize mutations in general in cancer. But I would say that this general approach, however it was comprehensive at the time, this general approach is now being utilized across cancer centers in the US and in the world in general, usually on more targeted panels against actionable genes. And so in these earlier studies, we basically utilized the study to really examine the landscape of alterations in metastatic cancers, cancers that have spread, and we looked at over 30 different metastases through this program, 30 different cancer metastases, as well as metastases taken from different sites, including those from lymph nodes, lung, and liver, bone metastases, and so forth. And this program, in that we were also characterizing the transcriptome, really gave us insights into the tumor microenvironment in addition to the tumor cells themselves. And in terms of the first 1,000 or so patients that we sequenced through these precision oncology efforts, we were able to see that patients that received sequencing-directed therapy, generally about 40% of individuals could receive a potential clinical benefit from understanding the molecular basis of their cancers, albeit these are all patients with metastatic or advanced cancer and highly enriched for patients that have relatively rare cancers in this first set of 1,000 patients that we sequenced. Through the MyOncoSeq program in general, we've had a number of intriguing clinical vignettes where we've actually learned from individual patients. For example, we discovered the NAB2 STAT6 fusion, which is a pathognomonic fusion for solitary fibrous tumors. We identified rare but targetable FGFR kinase fusions across an array of different epithelial tumor types. We identified the ER or ESR1 mutations as a common resistance mechanism in ER-positive metastatic breast cancers, generally patients treated with aromatase inhibitors, as well as in the context of prostate cancer. As part of a Stand Up to Cancer dream team, we found that in metastatic castration-resistant prostate cancers, approximately a quarter of patients actually have defects in DNA repair-related genes, such that these are genes such as BRCA1 and BRCA2, such that these patients could benefit from PARP inhibitor therapy, and PARP inhibitor therapy is now an FDA-approved therapy for prostate cancer patients with these DNA repair defects. The last two vignettes that I'll mention, this one's actually related to what Ben presented earlier, around this gene CDK12 that we discovered as being biallelically altered in about 6 to 7% of metastatic castration-resistant prostate cancer. And we discovered this subtype of prostate cancer, a relatively rare subtype of prostate cancer, because this subtype of prostate cancer harbored the highest level of gene fusions. And these are generally, most of these gene fusions are passenger gene fusions and not necessarily driver gene fusions. Had a much higher gene fusion burden than other molecular subtypes of prostate cancer. This is exemplified here by these red hashes in CDK12 mutant prostate cancer versus other subtypes of the disease. And the general mechanism that we characterized for CDK12 was that in metastatic prostate cancer, we see a biallelic loss of CDK12. This leads to genomic instability and extensive focal tandem duplications that occur in the genome. This then causes gene fusions or genes to go out of frame and that create neo-open reading frames or neoantigens that potentially activate the immune system in these patients that have CDK12 biallelic loss. And this was quite interesting, and we have ongoing clinical trials to try to extend these findings. Prostate cancer in general do not respond to immunotherapy. However, small subsets of metastatic prostate cancer patients do respond to immune checkpoint therapy. And this is an example of a patient that has biallelic loss of CDK12, where we were able to treat this patient with pembrolizumab, and you can see a massive decrease in PSA levels. The immune infiltration, these are T cells colored in brown here, presumably caused by the gene fusions and neo-open reading frames caused by loss of CDK12. This is an imaging study of a extrailiac lymph node, PSA of 8.9, after 13 cycles of pembrolizumab, decreased diminishment of that lymph node, as well as PSA dropped to 0.2. And in this particular patient, this was a durable response. And we're beginning to really expand this into identifying this rare subtype of prostate cancer that has biallelic loss of CDK12 and has a high gene fusion burden, to see if they might be susceptible to immunotherapy. The last anecdote in terms of clinical sequencing that we discovered was around the characterization of FOXA1, which is a pioneer transcription factor that appears to be mutated in a mutually exclusive set of prostate cancers that do not harbor ETS gene fusions. We actually classified the types of alterations that occur in terms of the FOXA1 gene itself, and upwards of 35% of prostate cancers harbor an alteration of FOXA1. And as I had mentioned to you earlier, these are mutually exclusive with the ETS fusions. But perhaps the most intriguing family of alteration that we identified were class 3 alterations, which create tandem duplications and gene fusions that occur in the FOXA1 locus that then lead to ectopic overexpression or very high levels of overexpression of FOXA1 in prostate cancer. And what I'd like to end with is a more recent story. As I had mentioned to you perhaps earlier, the gene fusions that we identified in prostate cancer create overexpression of an oncogenic transcription factor. Transcription factors in general have been thought to be an undruggable class of molecule. And so one of the approaches that we've explored is, can we find ways to indirectly target these oncogenic transcription factors by impeding their chromatin accessibility, or impeding their ability to bind to cis-regulated enhancers in terms of driving oncogenesis. And in prostate cancer is actually a transcription factor-driven disease, which I've listed here. You've heard about AR, FOXA1, MYC, as well as ERG, are all important drivers of prostate cancer development. And in order to understand the interactomes of these oncogenic transcription factors, we ended up intersecting some data that we had generated in our lab, as well as from publicly available data. And when we intersected these interactomes of AR, FOXA1, and ERG, the proteins that were common across these oncogenic transcription factors were components of the SWI/SNF complex shown here. A little bit of background in terms of the SWI/SNF complex in general, also known as a BAF complex. It's a multi-protein complex shown here. It mediates nucleosomal repositioning, and it does so through its ATPase domain, in terms of it employs ATP hydrolysis to disrupt these histone DNA contacts. The ATPase subunits are SMARCA2, also known as BRM, and SMARCA4, BRG1. And SWI/SNF is actually, this complex is mutated in upwards of 20% of cancers, but is rarely mutated in prostate cancer. And to probe the biology around the SWI/SNF complex in terms of these oncogenic transcription factors, we took advantage of developing a PROTAC degrader against SWI/SNF, and you heard again from Ben in terms of that as being a specific degradation technology, whereby we're utilizing a targeting moiety against the bromodomain of the SWI/SNF ATPases, shown here. It has a linker region and an E3 ligase ligand that can then engage the proteasomal degradation machinery, such that you can specifically degrade these SWI/SNF ATPase components at relatively low concentrations with this probe compound directed again against the SWI/SNF ATPase components. And one of the observations that Lambo Zhao made as a postdoctoral fellow at the time in my laboratory was that cancers that were driven by oncogenic transcription factors, such as prostate cancer, which is again driven by AR, ERG, and FOXA1, shown in red, were exquisitely sensitive to the SWI/SNF ATPase degrader relative to most cancers, as well as benign cell lines shown here. And this was quite remarkable in that AR-negative prostate cancer cell lines were relatively resistant to these SWI/SNF ATPase degraders. And so just to review, it's really, in terms of the cell line screen, we noticed that generally cell lines that were, or cancer cell lines that were driven by oncogenic transcription factors, including prostate cancer, as well as subsets of multiple myeloma, which you heard was driven by transcription factors, were quite sensitive to the SWI/SNF ATPase degraders. To really assess essentially how these SWI/SNF ATPase degraders work, we took advantage of ATAC-seq to look at chromosomal accessibility, and one of the first things we noticed was a massive chromatin compaction that was occurring upon incubation with these SWI/SNF degraders, shown here, and this was in sharp contrast to the BRD4 degraders. And what's interesting is that these sites that get condensed in terms of chromatin condensation generally harbor cis-regulated enhancer elements, and that the areas that are actually spared are areas that harbor promoter regions, and it's generally areas that harbor cis-regulated enhancers are generally in intergenic regions or in introns that get rapidly and completely compacted with the SWI/SNF ATPase degraders. And when we look at the consensus motifs at these chromatin compacted sites, we see motifs that match FOXA1, AR, ETS transcription factors, and so forth that get compacted in the context of these SWI/SNF ATPase degraders. When we look at the oncogenic transcription factors directly, we see that they get decommissioned from these chromatin compacted sites. This is by ChIP-seq analyses. Against AR, you see that AR is evicted from chromatin, FOXA1, as well as ERG, upon incubation with the SWI/SNF ATPase degrader. We also see diminishment of the H3K27 acetylation mark, which is a mark of active enhancers. So these oncogenic transcription factors are being displaced from these active enhancers when the chromatin is condensed in the context of these SWI/SNF ATPase degraders. To begin to look at these interactions in a three-dimensional space, we took advantage of high-C or high-ChIP-seq analyses. Here, carrying out high-ChIP-seq analysis on the H3K4 trimethylation mark, which is a mark of active promoters. And here I'm just using a exemplar of the AR promoter element, and we're basically using this high-ChIP-seq technology to essentially, in a three-dimensional view, examine contacts between promoters and enhancers. And as you can see, when we pull down H3K4 trimethylation and we pull down this promoter of AR shown here, we can map interactions to the cis-regulated enhancers. And this is a contact matrices showing these interactions between the AR promoter and the cis-regulated enhancers. However, when we incubate these cells with the SWI/SNF ATPase degraders, these contacts are markedly attenuated. So these enhancer promoter loops are lost in the context of the SWI/SNF ATPase degraders. The genetic programs emanating from these oncogenic transcription factors, whether they be AR, ERG, FOXA1, or MYC, are markedly inhibited with the SWI/SNF ATPase degraders. And when we begin to use preclinical models examining these SWI/SNF ATPase degraders in models of AR-positive castration-resistant prostate cancer, we were able to show, first off, that AR antagonists such as enzalutamide could inhibit tumor growth modestly. The SWI/SNF ATPase degraders themselves, as a monotherapy, further inhibited tumor growth. However, when we combine the SWI/SNF ATPase degraders with the AR antagonists, we actually saw tumor regression in these models represented by, for example, this waterfall plot with the combination therapy. And this was quite remarkable because we were seeing this preferential impact on these oncogenically driven tumors. However, we're not seeing toxicity in these model systems. While we're seeing nice degradation of the SWI/SNF ATPase components, we did not see changes in body weight, organ weight, complete blood count, or histological changes in the normal tissue of these mice. So then this brings us to this general model in terms of this general idea of targeting oncogenic transcription factors by basically impeding their accessibility to these cis-regulated enhancer elements, where essentially in the oncogenic state, you have this heavy enhancer promoter looping that drives supraphysiologic expression of target genes mediated by these oncogenic transcription factors. When you come in and degrade these SWI/SNF ATPases, this then leads to heterochromatinization of these cis-regulated enhancer elements, eviction of these oncogenic transcription factors, and then you're left with sort of basal expression emanating from the core promoters, likely attributing to the fact that normal cells are relatively resistant to these SWI/SNF ATPase degraders relative to cancer cells, which are addicted to this oncogenic transcription factor activity and dependent on these enhancer promoter loops to drive supraphysiologic expression of these target genes, thus explaining why we're seeing preferential effects in cancers that are dependent on oncogenic transcription factor regulated growth. With that, I'd like to thank the individuals that were a part of my team in terms of this particular more recent study, including Lambo Zhao, Abjit Parolia, and Yuan Chao. They were co-leaders of this study, as well as a number of other collaborators. Of course, like to thank our current lab, including current trainees, as well as a number of individuals in our group, our lab manager, who's really helped in terms of guiding a number of these studies and coordinating a number of activities in the lab, Shuhong Chao. The original gene fusion team, represented by a number of graduate students at the time, as well as those that have gone on to become faculty, a number of postdoctoral fellows, as well as collaborators, including Mark Rubin, Ken Pienta, and others, and the two graduate students at the time, Scott Talmans and Dan Rhodes, for the early work in terms of that project. And then finally, I'd like to thank my family for their gratitude and support over the years that have really allowed me to work on these studies. And as you heard from Michael, my family is also dominated by females, including our friendly pooch as well. With that, thank you very much.